None of the content on this or any episode of the Kratom Science Podcast, Kratom Science Journal Club, or on any page of KratomScience.com is intended, nor should it be considered, medical claims or medical advice. This is the Kratom Science Journal Club with Dr. Jonathan Cachet, neuroscientist and expert in psychopharmacology. In each episode, we discuss an article in a peer-reviewed journal. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. Cannabis Museum Community Hemp Fair, Saturday, April 22nd, 1 p.m. to 6 p.m. Live music, hemp speakers. Does that mean the actual sound system and the speakers is made out of hemp, or there's going to be people speaking about hemp, or they're going to be like hemp people, like scarecrows, straw men speaking as... yeah.
I think it's probably yes to all of those except for the hemp speaker one. I've yet to, unless Henry Ford developed those alongside his car made out of hemp. But we've got some hemp farmers coming in. We've got some other folks in the hemp industry. We're going to have live spinning of like fiber hemp into cordage. And all of that's to sort of celebrate. So the museum just opened in its new location in Athens, Ohio. We have hours Wednesday through Saturday, 12 to 5, when you can generally swing through. And the current exhibit that's up now is called Hemp to Hackles. And so it's a bunch of old farm equipment and cordage and like different ropes, different clothes, like all the stuff that you can make out of hemp. But it starts with the hemp field. And you sort of, as you walk through the exhibit, you go through the different stages of processing it, and by the time you end up at the end, you get a smorgasbord of like collector's items on rope and hemp clothing and all the different uses there. And in fact, in the gift shop, we have some hemp clothing available as well. So yeah, we're pretty excited about that.
Awesome. I think we'll come down to that. I think that's when we will visit
that would be the time.
The Cannabis Museum.
Fantastic. I know my wife is busy planning. They're there every week, calling vendors. There's going to be food there and some food trucks.
Awesome.
And then I just got asked this morning if I could MC the event, in between all of the different speakers and panels and stuff. So
cool.
It should be a good time.
Let's get into it. So this is our 40th episode, so I thought we'd do one of your old papers. This is from 2010, and it's Modeling Withdrawal Syndrome in Zebrafish. It's from Behavioral Brain Research. Big team from Tulane University Medical School in New Orleans, where I was about this time last year.
Fantastic. Yeah. Did you go down there for Mardi Gras? Mardi Gras just ended.
No, we went for St. Patrick's Day. So it was like mini Mardi Gras. There was only 4 parades while we were there.
Irish Channel, cabbage in the streets.
Yeah.
I've been there.
We were there. Yeah, that was fun. It was really fun. I've never been to New Orleans before, but so how long did you live down there?
I was there for 5 years, 4 years for grad school, but then I did a little stint in Southern California, but that was also my first time down there. I had just finished undergrad and was looking for graduate school opportunities where I wanted to get into something that was psychopharm, so drugs, brain, and behavior. And essentially, there was a call for graduate students to help set up a new Zebrafish psychopharmacology lab down at Tulane University in New Orleans. And if it, then I called, interviewed. It was like, okay, this is behavioral research focused on the nervous system that involves controlled substances, and all the people were cool. And I'm like, I'm in. New Orleans wasn't even on my radar at that point. And to be honest with you about it, I haven't lived down there for at least 10 years, but the background on my phone is still Live Oaks in the street of New Orleans. They say once you're there, if you're there and then you leave, you sort of always long to come back.
Hell yeah, that's our experience. It was fun. And we did everything. We went on that street with the Live Oaks. We took the streetcars. We're going to San Francisco this year. There it's cable cars. In New Orleans it's street cars. And Pittsburgh used to have trolleys. I wish we still had them. Some people call the above ground trains, trolleys too. But they're not really trolleys. It's funny because they have the whole, like, get the money off the tourist culture there. People, they say, where'd you get your shoes? And if you tell them, they say, give me money. I don't even know how that works. But
on your feet. You got them on your feet.
Yeah. the scam that is relative to this podcast was when we went down last year. Like, nobody knows if weed's legal from one state to the other now. So, like, people will book a vacation there and be like, oh, I guess weed might be legal in Louisiana. It's not. So they land, hemp is legal and CBD is legal, and hemp flower rolled into joints. So they'll have, like, trucks on the street that say pre-rolls, and they'll have big marijuana flowers and, like, Scooby-Doo with stoned eyes, and they'll be selling pre-rolls, but people are like, oh yeah, hook me up. And meanwhile, they're getting some CBD hemp there. So
for sure, yeah. That, even before all the medical stuff took off, like, even 2010 when I was down there, they had that same truck, but they were giving out suckers that were essentially like cannabis flavored, but there was no THC in there,
they have that too. Edibles, they call them edibles, but
if
they don't tell you what's in them.
The French Quarter, be leery, right? Especially if you're an out-of-towner, because all of the stores, like, if you're gonna buy beer before you go in the French Quarter, it's one of the best things about New Orleans. If you're going out drinking, you can just go to a gas station, get a 12-pack, and walk down the sidewalk with your own 12-pack rather than paying the elevated prices at the bars. But if you buy it on Canal Street, it's for sure like $5 to $7 more expensive than if you just went into downtown, maybe 2 or 3 blocks, and bought from a gas station there.
There's a way to live cheaply.
Also, another pro tip: the best fried chicken down in that area is from Brothers, the gas station.
Brothers, the gas stations. That's good to know. I was gonna ask where the best gumbo was.
One of my favorite restaurants, and it was called Dante's. It was uptown where Tulane is, but I know for sure that restaurant's not there anymore. They turn over so quickly. So, like, frankly, I would be surprised if 15% of the restaurants that were there when I were there are still around. Like, it's generally the service industry like that turns over quickly, but in New Orleans, even quicker.
I got it at Antoine's, it was like our fancy lunch. And we were down there eating it, and all of a sudden we look over. Tommy Lee Jones walks through the door with, like, 2 younger dudes that I think there's a movie being filmed through there, and he went up. They have a whole section for celebrities, so he went up in the celebrity section, and the young waiter was like, "You know who that was?" "Yep, the guy from Men in Black." I was like, Men in Black? I've known him from way longer than that. What about The Fugitive? That is
Nicolas Cage, John Goodman, all sort of like just random.
Yeah,
they just show up somewhere.
Nick Cage was there, and they did like a cartoon of him in the paper of like him drunk or something, because he has these weird grave down there that looks like a pyramid. That it's his grave that he already bought. So that's interesting. Anyway, that was only one small part of that study. So for this, so you studied diazepam, ethanol, morphine, and caffeine.
Yes.
That's the 4. Yes. So did you have to get a DEA license for morphine?
Yes. We had to get a DEA license for all of this. Yep.
So what was the process of obtaining a DEA license like to do this study?
A good question. So the principal investigator of the lab is a Russian citizen who moved to the U.S., and I thought that might entangle things a little bit, but essentially we filled out paperwork, did background checks. You have to get the university, the head of your department to sign off on it. You're in the Department of Pharmacology. You have to provide them with the detailed protocols, all the steps of the experiments that you're doing, and what you're doing them in, how quickly you think you're going to be moving through, what kind of estimates on the amounts that you're going to need. It's really essentially the same sort of information that you would put in a grant if you were writing to get research money from National Institute of Drug Abuse or some of the other NIH things. So, really we could take the grants that we had already written and got funded to a certain degree, put that into this paperwork, and then you send the packet off to the DEA. You also have to describe your protocols for storing, disposing, and handling the controlled substances. And so we had to get get the locks changed on all of our lab doors. We had to get fireproof safes. We had to I remember making inventory sheets. Of course, the PI's name was on all of this, but as the lead graduate student in the lab, I was the one that was actually in charge of actually moving and doing all this stuff. But especially as you notice too, I don't know exactly how many folks are on this particular paper, but we had a lot of people cycle through that lab. A lot of maybe 40% of these folks are undergraduates that were in Tulane's neuroscience program. Some of them are medical students that were in Tulane's medical school. Some of them were graduate students that were in the same program that I was in. Either they were going for their master's or their graduate degree. But we had to do things only the PI and the lead graduate student are able to access the safe and weigh things out and then give it to people. And a lot of times too, the folks that were running the experiments would come in and there would be 4 vials on the workbench, with water in them. And it would just be A, B, and C. And some of those would have nothing in it. And then 2 of them would have different, ratios of what a drug concentration, and you couldn't tell. So they were blinded from what was what. It was actually easier and less road bumps than I thought to actually get the license from the DEA to then, you get basically a number, as if a doctor has a number for a prescription pad that you use, and you get these special forms for ordering controlled substances from chemical companies like Sigma Aldrich. And that was it. It was paperwork and demonstrating that you can handle, or you're putting in proper protocols in order to handle, they say controlled substances, and it's to a certain degree they treat it as if it's nuclear waste, but it's what you got to do.
So how many people are granted a DEA license per year? How many scientific teams around the country? Do you know? Ballpark?
I don't know off the top of my head. It's different too. So there are a few research labs that were in the pharmacology department at Tulane that were investigating safe alternatives to opiates, right? There's Dr. Zadina's lab, they call it the Holy Grail, but it's you get pain relief and you don't have that addiction potential, which I think is a myth, right? You're sort of chasing a myth there if it's an opiate. But that's a controlled substance. So they had the license for that. And other labs, I don't think lidocaine isn't a controlled substance. It may or not be, but essentially the rare part that we got was access to Schedule 1. So that's basically LSD, PCP, psilocybin, cannabis, all of the level 1s. There are a much lower number of labs that have access to do research with the controlled substances. And, if I had to guess, I would say less than 50 in the US, and maybe that's changed, but that's just my gut feeling on it. It's not necessarily the hard fact that I've seen any statistics from the NIH. I think it's increased, but I know that it's not very big, because when you're doing this type of scientific research, you of course go to conferences and talk to other groups, and you review the papers of other groups that are doing things similar to you. And we were really the only ones in the adult zebrafish behavior space that had access to these type of controlled substances.
I mean, was that just for the morphine that you had to do that, or the diazepam? What's diazepam?
It's a benzodiazepine.
Oh, okay, yeah. So was it those 2 you had to do, since ethanol and caffeine are both legal substances?
Yes. And those are just the ones that are presented in this paper. There are, I think I've got 46 papers or so that came out of this lab, and there's LSD, MDMA, psilocybin or psilocin. So of the ones that are just in this particular withdrawal study, it was just those. It would just be those 2.
Does DEA do a lot of random inspections and stuff like that?
No, I don't think we were ever randomly inspected.
Okay.
I've had enough experience with regulators everything's good if everything's just sort of hunky-dory status quo. Like if something were to happen and something were to go missing, or somebody was to get arrested and then say, something like that, then they'd start coming down. But we never had any issues like that.
It seems like one of the goals was to establish that zebrafish could actually be a legitimate way to study psychoactive drug effects. Was that actually one of the goals?
Exactly. You nailed it right on the head. So like zebrafish were established as an animal model in genetics research. And there's a number of nice aspects about zebrafish, like namely their embryonic development. Their bodies are clear for like quite a bit of it in the beginning. And so a lot of the genetic manipulations, like Sonic the Hedgehog or the hedgehog genes that control when genes get shut off and this turns into a heart, or genes get shut off and these turn into skin or skin cells or scales. A lot of those were discovered in zebrafish. So there was a foundation of using zebrafish in scientific research, particularly from a genetic perspective. So we sort of okay, that's cool. Let's keep that going. But then the question was, what about their behaviors, and how can we quantify their behavior, and how does that even relate to behavioral phenomenon like drug withdrawal? And so this was one of the, not the first, there was one other group up in Canada that I think did ethanol withdrawal and had published on that prior than what we did. And so we were able to look and see what they did with the ethanol prior and say, okay, well, let's try this with a bunch of other drugs. And really the goal of the entire lab was to set a foundation down of this is what withdrawal looks like in zebrafish, both behaviorally and physiologically. And then all of the other ones, this is what psychedelic, this is what psychedelic drugs do. This is what anxiolytic drugs do. So this is when zebrafish are not stressed. This is when zebrafish are stressed. And so, my whole dissertation was essentially on, can we do that? Is it viable? And can we match the behavior and the physiology? And what do we see? So how do we make a behavioral catalog of how adult zebrafish behave in these different conditions? The other one too is we did social behavior, and we did MDMA studies. So fish in general, they get together in schools, and how tight the schools are. So the distance between all of the fish in the school was a proxy for whether or not it affected social behaviors or not.
Oh, that's cool. They're still using zebrafish in a lot of studies, right?
Yeah, and there's been a bunch more labs since our work that have essentially picked up the 3D tracking and machine learning, swim trajectory methods that I had developed then, that are now using those worldwide.
That's awesome. Did you do one in Kratom at all?
We did. We brewed Kratom in giant, gallon scientific vats on a hot plate with a stir rod, which is frankly the best way to brew tea, if you ask me. One of the things that we sort of realized as we were about to put the fish in, so we'd essentially have a water, and then we'd have a little bit of Kratom, a lot of Kratom, and then we'd put the fish in, you drop them in the tank, and then monitor their behavior. But because of the tea, the fish that were in the Kratom liquid were in green water, and the control was in clear water. And we had to compensate for the differences in color, because we didn't, we wanted to eliminate the potential influence that could have on their behavior. And so what we ended up doing was putting them in there and then turning off the lights completely, so they were just in the dark. We probably did it, with, let's say, anywhere from 15 to 25 fish. And we did it probably, 5 or 6 times. There wasn't a lot of great data that came out of it, or there weren't a lot of significant differences between the control and the treated fish. And I don't think that it changed their cortisol levels, so their physiology. And I think we also did some where, they were exposed, just like in this withdrawal paper that we're looking at, you put zebrafish in a liquid that has morphine in it. 2 weeks they're in there. So they're getting used to the morphine that's in the water, and you keep the morphine levels up and high. And then to induce sort of withdrawal, you pull them out. And then sometimes we would pull them out and give them naloxone. So it was an immediate withdrawal. We did maybe once or twice pull them out after chronic treatment in morphine and put them in Kratom. the hypothesis was it would help, and it would probably reduce the amount of crazy swimming they're doing. It would reduce the amount of cortisol that their body released, so they would be less stressed than versus the ones that just get forced into withdrawal. But ultimately, again, I think like we saw trends that looked that way, but it wasn't, p-value 0.05 statistically significant. So it never ended up making it to a publication.
Yeah, I was just looking up on PubMed. There's 2 Kratom studies on zebrafish. One just came out in 2021. I don't even think I even looked at that one. It's about toxicity assessment. I think at one point you told me the only thing that blew up their hearts was caffeine.
Yes. Yes, that was the biggest one. So and it looks like that's in embryos, right? So you can sort of see in the photos there, you can see how the embryos are somewhat clear and how it affects. So they're looking at does kratom affect the development of the different body systems? So this is more along the lines of what they traditionally did with zebrafish, where we were specifically looking at zebrafish that were adults, which were essentially 2 to 5 months old. Yes. So because no one had developed or given these controlled substances to zebrafish before, adult zebrafish,
yeah,
we had to establish, just like this comparative toxology experiment, we had to establish at what dose does it start affecting or modifying their behavior, and at what dose does it become lethal?
Yeah.
And the very first time we did this, we were dumping in controlled substances like they were fish food from the top. And it would just be little, you can imagine a little cloud falling, but it's a little ball that's falling in. And then all of a sudden, of course, the zebrafish just munch it and eat it like it's food. So they're getting a huge dose of morphine. And so we realized very quickly that before we put it in the tank that the fish are in, we either need to mix it in the water and then add the fish, or we need to mix the powder in a smaller vial with water and then pour that water into the tank. And so, we ran these studies. Every time we wanted to do a new drug, we would figure out at what dose is the threshold dose where we can start noticing changes in their behavior, and at what dose does it become lethal. And, we were pretty, we weren't it's not like we were killing hundreds of thousands of fish to figure this out. Maybe we did 5, and then we got an average about where around a lethal dose would be. And we could use that information to say okay, this would be a low dose, this would be a medium dose, this would be a high dose. And then, of course, several milligrams per liter higher than that would be the lethal dose.
Yeah.
But of all the drugs we did, for sure, caffeine was the most alarming in that I wasn't expecting to get to some point, and then the zebrafish literally, so you could see its belly explode, it flips upside down, and it's dead. And you're not expecting that with caffeine. You're oh, it's just caffeine, c'est la vie, let's just scoop it in there. You're expecting it with morphine or ethanol at some point, but gosh, man, caffeine really took us all by surprise. And the fish, as soon as they got over a certain dose, it was lights out. The only other thing that's sort of similar to that, and then maybe it's not my proudest moment, but at one time we were curious what would happen if we put zebrafish in Red Bull, and they also immediately died. And I don't think it was because of the caffeine per se. I think it might have had something to do with the carbonation and the way that interacts with their gills. But again, you expect it to be all right. It'll we'll put it in there for a minute, and if it starts looking in distress, we'll pull it out. But these are guys just within 5, 6, 7 seconds, they're dead.
I got a new reincarnation fantasy. I think a zebrafish in an LD50 study, but which substance? Maybe morphine and pour a little bit of vodka in there or something. That'd be perfect.
Yeah. Well, now you don't want to be in a lethal dose study. You want to be in an effective dose study.
Yeah, but 4 to 6 weeks as a zebrafish, and then you get reincarnated as something else.
Oh, okay. I got you. Yeah, it's just a quick in, quick out. Insy outsies. I like it.
So what types of behaviors are you looking for once you give them the drugs, once they're in drug-infested waters?
So the group that was before us, they called it a novel tank, and it's analogous to what rodents would use with a novel environment. But essentially, the theory is if you throw an animal into a new environment that they've never been in, they will sort of get to a place where they feel somewhat safe or protective. And then over time, they'll gradually start exploring that environment more. For rodents, that materializes in, usually you put a rodent in a new environment, it'll stick against a wall or go to a corner. So like two of its, behind it is blocked by the walls. They'll look out there, and then eventually they might start running along the walls. And it's not till like maybe 10 minutes into the experiment, they'll do a cross from the middle, like right across the middle or the center of the table. And so the same with zebrafish. The presumption is that from an evolutionary perspective, one of the most dangerous places for a fish to be is at the surface, where they could get, scooped up from birds or other predators that are there. And so when you drop a zebrafish in a new environment, our tests were usually about 7 minutes long, 7 to 10 minutes long, but they would go to the bottom. And for the first 2 minutes or so, they'd stay at the bottom. Then maybe they'd do like a loop, and they would cross the halfway point that we had like demarcated for the people that were watching. Okay, so they were the profile becomes they were at the bottom frozen for 2 minutes, and they made one transition to the top, and then they go back down. And eventually at some point, they are going up to the top much more frequently. They're spending time at the top. And so that was the primary endpoint that we could start from, where, time spent on bottom, time spent frozen versus swimming, and then time spent at the surface. Other behaviors, and they became like challenging to define, especially between different folks in the lab, but it was called an erratic movement. When you see it, you know what it is, but it's hard to, define objectively, like quantitatively. But they would they'd be frozen at the bottom, just sitting there. And then all of a sudden, like do crazy darting behavior, looping all around, maybe it lasts 2 or 3 seconds, and then they're back at the bottom frozen. So we would call that an erratic movement. And really, when I got there and as we're developing these models, we were scoring all of that, where we had 2 people with clipboards, and they were literally tallying like up at the top, down at the bottom, up at the top, down at the bottom, erratic movement. And my contribution to help this lab was to automate that with video tracking software. And then once that hit, like the notion of an erratic movement could be quantified as, we had all sorts of new variables available to us. What's their velocity? What are their turn angles? So an erratic movement becomes a sudden jump in swimming velocity with tight turn angles. And so that's really what led to the machine learning. Like once we did video tracking software and we had all sorts of those movement variables, movement patterns, it really expanded the amount of differentiation we could see between different substances. Like it's very clear when they're stressed out versus when they're not stressed out. It's like the diazepam that we had in here. You give them a diazepam, let them sit in it for 2 minutes, then put them in the novel environment, and they don't even go to the bottom. They're just chilling up top, like just hanging up there, swimming along the top. But then, you get to withdrawal, you get to social behavior, you get to the psychedelics. Just quantifying the top versus the bottom and amount of time frozen or not frozen wasn't really enough to differentiate between the different psychedelics and the neurotransmitter systems they hit. And so the video tracking really helped with that.
Okay, so once you took them out and you put them in clean water, so how do you measure withdrawal in zebrafish? Is that the cortisol, or is it also have to do with movements and stuff?
Also has to do with movements. So the cortisol was a physiological measure. So after they were done swimming in the novel tank for 7 to 10 minutes, we would then take them out, euthanize them, and put them in like little test tubes, and usually into like a -80 freezer. And then there would be a bunch of extraction steps that you would do to just get the amount of cortisol that they had in their whole body. And then we could quantify that. So that was a really good way to say, okay, behavior looks like they're more stressed out. I'll get into the details there. But then when you can say that, but you run the risk of anthropomorphizing. Like, is the zebrafish really stressed out? We can't ask it. It looks like it may be stressed out, but we might just be, that might just be our perception. So the value of having that physiological measure as well, or you could say they swam like this, which was kind of crazy. They had a lot of erratic movements, and their cortisol was 2 or 3 times higher than the fish that didn't get the drug treatment. That was a pretty good combination. And so for drug withdrawal, essentially we are treating like the home tank of, let's say, 20 zebrafish with, let's see what the dose was, 1 milligram per liter of morphine. It looks like chronic morphine 1 week. So For a whole week, they were in a tank, in their home tank, with 1 milligram per liter of morphine. When we take them out, we either give them naloxone or just put them directly into the novel tank. And what we'd expect is, like, if they were fine and just cruising, like if they just had a short dose of morphine, they would be swimming at the top. They would not be, they would have much more transitions from the top to the bottom. They would spend less time freezing. There would be less erratic movements. That's what we see with short term. If we go long term, then cause withdrawal, it's the exact opposite. They're frozen at the bottom for the 3 minutes. Then they do like a 7-second erratic movement, followed by a 3-second freeze, followed by another erratic movement. So how many times did they sit frozen for a little bit? How many times did they go through the erratic movements? And you can see that, like, with the erratic movements in particular, like if you look at the, what is this, would be 2A, the amount of erratic movements was significantly higher between the control fish that were not exposed to drugs, the chronic drug exposed drugs. So with those, we just took them out of the tank and put them in. And so, like, within the first 7 minutes, we wouldn't expect them to go through withdrawal. But then with the draw fish, we hit them with naloxone immediately and then put them in the tank. And that was what induced the withdrawal.
It's interesting, the similarity between the fish and how mammals would react. Do biologists look at this and use that kind of for what they study in human behavior and mammalian behavior, fish behavior, a kind of a comparative understanding of how, like, a substance would affect?
Yeah. Both, so translational. Like, is it translational between the different species? And so, like, of course, they already had thousands of studies where they were looking at morphine withdrawal in mice. And so, with the open field test that I was mentioning earlier, you sort of get the same profile. Like, they're not crossing the middle, they're frozen the whole time. And so, that aligned well. And with zebrafish, yes, I mean, there is translational value there. And so, like, where the genetics came into play is they have all of the same. They have 2 dopamine receptors. They have all 3 of the morphine receptors. And so then we could, like, knock out a morphine receptor and see if they still go through the same degree of withdrawal. Or, like, in the lab down the hall from us, you could make new opiates and have that as a powder and then treat them for 2 weeks and then induce withdrawal with naloxone and see if they are just as stressed out based on their erratic swimming and or their cortisol levels or not, right? And so the value of the zebrafish in a translational perspective, like, it allows you to screen 100 candidate novel opiate structures for the ones that induce the least amount of withdrawal behaviors and physiological response. And then you can narrow that down to, like, okay, here were the 5 that didn't seem to cause that bad of withdrawal. And you take those into mice and you just keep on moving up the chain, the species chain.
I just watched this piece on drug use in the animal kingdom called Cocaine Bear. So I
saw that and I had no idea what it was.
It's a great movie. We went to the theater and saw it. It's hilarious. But I'm talking about drug use in the animal kingdom. Like, we know, like, dolphins can eat pufferfish and get high. And I'm sure this has been covered on Joe Rogan, but is it mostly intelligent animals that would do that, like, on purpose? Or, like, would zebrafish in the ocean find, like, a fermented piece of coral or something and keep eating it for any reason whatsoever?
Not zebrafish.
Yeah,
probably not. They're in rivers, like in primarily of Southeast Asia. And so I don't know what they would go for. Like, when it comes to the sea animals, I would think much higher order, or like maybe octopi. But I was going to mention, like, the fermented apples. Like, I think everybody's seen the video on YouTube where essentially, like, hyenas to elephants to giraffes to, like, almost every animal on, in the Serengeti, they know that if they come to this particular tree after the fruits had fallen off and given some time to rot, that they ferment, and then essentially they all just gorge themselves and then pass out after wobbling around for a little bit. So, they certainly do have, and I don't want to say it's not like drug-seeking behavior. I would always classify it as, like, we're interested in modifying the sort of parameters of our perception and just changing that, I think, in order to understand it a little bit more. And, maybe it's the same for some of the other animals.
Cocaine Bear, probably, his mouth probably got numb and it felt good. So he's just like, All right.
Well, yeah, with cocaine, like with all those drugs that hit the dopamine, essentially like it's the same pathway that you get when you have sex, right? So they're like, oh, wow, that felt really good. I'm going to do that again.
And there was a cocaine bear in real life, but they just found him dead with a bunch of chewed open kilos of cocaine.
Well, and so what? Yeah, so is that what they found, or was there like
that was real life
repeated use?
I think he just ate until his heart exploded. Because the beginning of the movie is nonfiction. It was a guy was paranoid that he was being followed by the DEA or something, so he dropped a bunch of cocaine out of a plane. He had a Cessna or something, and then he took a bunch with him and he parachuted into the woods. And he was going to run away, but he was too weighed down. This is real life. He was too weighed down by that cocaine, and the parachute, he descended too fast, and he died, and he ended up in the middle of the woods. But there was plenty of cocaine in the forest, and they still haven't recovered it all, but they found a dead black bear with a bunch of chewed open packages of cocaine around it, and they took the blood test. I was just reading the article, and it was 4 grams of cocaine in its blood, in the bloodstream.
Wow. Yeah, that's
so it had eaten something they estimated, think about it, 30 kilos or something like that. Anybody listening can look it up and correct us in the comments, but so that's what happened. But the movie is great. The movie is, I think the guy who wrote it is it's my twisted fantasy on what would happen if a bear ate cocaine in the woods. So it's hilarious. It's pretty funny.
And you would assume that it got into this stuff, felt good, and then just kept going until it overdosed, rather than he saved a little bit and came back the next day. Yeah. That would be wild.
Yeah. So how much cocaine does it take to kill a bear? It's, at least 30 kilos, but it would probably be less than that. But I think he just kept eating until he died. Anything else about the zebrafish?
Figuring out and laying the foundation of this withdrawal behavior, essentially what we were studying was anxiety and affective state, different, stress, high stress, low stress, antidepressants, all of that stuff. And coming from it from a psychopharmacology perspective, using withdrawal in order to understand how an animal behaves when it's stressed is just one angle in which you can approach their behavior, right? And so, okay, we're going to induce drug withdrawal, and now this is how they behave. And now we have a template for this is what a stressed out fish looks like. There's other ways to approach it. All fish have this thing called alarm pheromone, where if a fish gets eaten by another fish, it gets chewed up or it bites down on it and it ruptures their skin cells. And the skin cells release this thing called alarm pheromone. So we would do it where we would just put a zebrafish in the novel tank and we would have extracted alarm pheromone. And, the instant that alarm pheromone hit the water, they just started freaking out like crazy. You just see it. And it resembled what the withdrawal state was. So, this was, I'm glad you sort of picked this paper, in that, this was one of the very first ones that we studied. This was probably my freshman year of graduate school, but it was really the combination of all of these things coming together to where we could then move on and be more confident in, okay, let's now study novel antidepressants, or let's study novel anti-anxiety medicines. Because we knew what a stressed out fish was and what a not stressed out fish was, we're then able to make, inferences about all of these new chemical compounds. So, it was really, really important to get that foundation there. And, of course, we studied it, and we probably, we did the Kratom one, less than a dozen times with maybe 10 to 15 fish each. But the amount of times that we modeled withdrawal or did chronic treatments to then induce, withdrawal, that was in the hundreds. And it was the foundation of a lot more. We could then go from there and say, okay, let's induce withdrawal to have one stressed out fish and put it in a tank with a school of fish and see how that changes how close they come to the other fish. So is there a social separation because this one's going through withdrawal and it's a high anxiety state, or does having the other fish in there, does it get closer and sort of alleviate, keep cortisol levels down? So yeah, it was fun stuff. I look back fondly on it, especially being in New Orleans, figuring out, doing something that hadn't been done before, especially when it involves giving fish drugs. It was a good time.
All right. Thank you, Dr. John Cachet, for 40 episodes, and you can meet him and me on April 22nd in Athens, Ohio at the Cannabis Museum Community Hemp Fair. Check us out on Twitter, Facebook, or on TikTok at KR80M Science. Please like, subscribe, rate, review, share it on social media. The music is Captain Big Wheel. The song is Moon Runner. Kratom Science Journal Club is produced by me, Brian Gallagher, for KratomScience.com. Take care.